the presentation we see there. It's very interesting for all of us, for me in particular, because it is about nanoparticles, silver nanoparticles, functionalized by using argon plasma grafting technology and enhancing their antibacterial properties, resulting in particles with a wide effect against gram positive, gram negative, as we are going to see, and also against the bacteria which became antibiotic resistant. So they have high specificity, biocompatibility, and stability. Dr. Traba is an associate professor on the Metropolitan Campus of the Fairleigh Dickinson University, is a published author, and also he makes presentations, and he is very good at writing successful grants. So let's listen to him. And again, thank you to everybody who is here today.
Hi, everyone. Thanks again for those very kind words. I really appreciate that. I also wanted to congratulate everyone today for their great work, and hopefully at our 23rd Student Annual Research Symposium, I hope to see some updates. With that being said, today my talk will be on new alternatives for the prevention of biomedical implant contaminations. Okay. In order to properly engineer antimicrobial or anti-infectious surfaces, several questions must be asked. The first is, what are infections? Next is, how do medical implants typically become infected? Followed by problems associated with medical implant infections. And then, what have others done? And finally, what is our idea? A biofilm is an infection, and it's an assemblage of bacteria irreversibly associated with a surface. It is a community of bacteria living together, encapsulated by an extracellular polymeric matrix, and is primarily composed of polysaccharides, proteins, minerals, metals, nucleic acids. Biofilms are 1,000 times more resistant to antibiotics and the host immune system in comparison to their planktonic counterparts. Biofilm formation begins with 1, the initial attachment of planktonic bacteria, followed by the irreversible attachment of these planktonic bacteria. Next is bacterial recruitment and reproduction, followed by the formation of a mature biofilm. And finally, if a biofilm is disturbed for any reason, planktonic bacteria are released, and this biofilm formation process happens again. Image 1 is a scanning electron microscope image of polycaprolactone microfibers contaminated with S. aureus bacteria. The scale bar for this image is 5 microns. In clinical settings, there are 3 primary pathways in which biomaterials and implanted devices typically become contaminated. The first is an open wound or an implanted device can undergo microbial exposure. The second involves the sanitation negligence of healthcare providers by not properly washing their hands. The third is native or transient bacteria of the human body, for example, S. aureus, have an extraordinary ability to colonize sterile foreign polymer surfaces. Image 1 is a scanning electron microscope image of a 1-day-old S. aureus biofilm, and the scale bar is 1 micron. Image 2 is another scanning electron microscope of 7-day-old S. aureus biofilm. The scale bar for image 2 is also 1 micron. So if I were to ask you the difference between image 1 and image 2, what would you say? You would probably say that image 2 has more stuff. And this stuff is the extracellular polymeric matrix. This ECM or EPM is what protects the biofilm from the host immune system, as well as from antibiotics. This ECM literally prevents the bacterial agents from penetrating the biofilm and resulting in bacterial death. S. aureus biofilm infections plague hospital settings and account for more than 70% of all hospital-acquired infections. These infections can form on biomaterials including cardiac pacemakers, joint replacements, catheters, and sometimes even tissues and organs. This is all because the solid-liquid interface between body fluids and biomaterials in vivo are ideal environments for biofilm formation. The contamination of biomaterials often results in functional impairment of the device, pain and discomfort for the patient, surgical replacement of the device, and sometimes can even result in death. Until recently, antibiotic immobilized surfaces were considered the most popular surface modification technique. In this technique, a high concentration of antibiotics are introduced onto a surface, and the antibacterial activity stems from a sustained release. Consequently, the bacteria are killed before they form biofilms. Some of the obstacles facing antibiotic immobilizations are: 1, these techniques lead to further bacterial resistance and spreading of contamination due to the suboptimal exposure of antibiotics, which simply means that doctors, physicians, researchers aren't providing the patient with enough antibiotics to completely destroy the infection. And 2, there is also a limited shelf life of the material due to the sustained or gradual release of antibacterial agents. And 3, they are not effective towards antibiotic-resistant bacteria, including methicillin-resistant Staph aureus, or you've probably heard of it referred to as MRSA. There have been many successful antibacterial surfaces, and these surfaces do not often use antibiotics. One of the most effective strategies involves the deposition of inorganic agents, such as metals or even ions, to a surface. These coatings are known to exhibit a broad spectrum of antibacterial activity. They have a gradual, persistent, and continuous release of silver ions, and this is all because of careful experimental design. And as a result, these materials also have a limited shelf life. And finally, they're often toxic to tissue cells and therefore are not suitable for implanted devices. The reason is the concentration of these antibacterial agents, these metals, is too high. So our idea is to develop a unique antibiotic-free anti-infection surface that can selectively kill bacteria and sustain tissue cell growth. Theoretically, we want our surfaces to be stable and biocompatible. We want to increase the shelf life. We want them to possess a long shelf life. We want to eliminate further resistance of bacteria to antibiotics by inducing a physical rather than chemical mechanism of action. And finally, we want our surfaces to remain effective against a wide range of bacterial strains. Figure 1 is a UV-Vis spectrum of silver nanoparticles synthesized throughout the study, where absorbance is on the y-axis and wave number is on the x-axis. We used the silver nitrate and butylamine approach to synthesize our nanoparticles. The absorbance from nanoparticles synthesized throughout this study was observed around 400 nanometers, and these results are in agreement with other published work. Image 2 is another SEM image of silver nanoparticles synthesized in this study. The scale bar is 50 nanometers. According to our SEM image, the average recorded size of the nanoparticles was roughly 25 nanometers, except for an occasional silver nanoparticle aggregate, which was found to be roughly 50 nanometers. The concentration of silver nanoparticle stock solution was calculated to be 5 millimolar after synthesis. With this being said, our primary goal was to synthesize extremely small nanoparticles. We wanted these nanoparticles to be extremely small because the smaller the nanoparticle, the more potent the antibacterial activity and the less toxicity towards tissue cells. In this slide, we see a schematic diagram of the Plasma Prep 3 device. This is our equipment that we used for our modifications. Plasma is a reactive cloud of ions, electrons, radicals, and neutral atomic species. A plasma can be produced by supplying energy to a gas in many different forms. For example, it can be electrical, optical, and thermal. Some of the gas molecules are raised from their ground state to an excited one, and this follows a modified electron distribution. An enhanced level of dissociation, excitation, ionization is thus accomplished, which creates new reactive species, including photons and charged particles. The plasmas generated in our lab are non-thermal plasmas. These plasmas typically have temperatures between 40 and 100 degrees Celsius. The advantages of using these cold plasmas are that the thermal load on the treated biomaterial is negligible, and the reactive species on the argon plasma are gentle, that surface modification is only achieved to a few nanometers. So that means that the surface of our biomaterial is still unchanged. This figure depicts an illustration of the plasma chamber. One is the Pyrex reaction chamber. Two is the lower semitubular electrode, followed by the upper semitubular electrode. Three is the gas inlet. Four is the radio frequency power supply. Five is our sample holder, which is approximately 8 centimeters away from our discharge gas inlet. And 6 is our vacuum connection. This figure illustrates the schematic diagram of the anti-infection fabrication process. In our research, an inert argon plasma is used and is gentle and does not alter the chemical composition of our biomaterial. In the first step of the modification process, the gentle argon plasma is applied directly to the substrate of our biomaterial. These biomaterials can be polyethylene terephthalate, or PET, or even silicon wafers. Plasma exposure is for 3 minutes and at a discharge power of 80 watts. These experimental conditions ensure uniform exposure to the biomaterial, and this activates our biomaterials. The activated biomaterials are then exposed to air for 75 minutes to generate our reactive centers. These reactive centers include hydroperoxide, hydroxyl, and peroxide groups. After exposure to air, we then expose our surfaces to 20% of acrylic acid monomer solutions. The graft polymerization reaction is carried out at 70 degrees Celsius under a nitrogen atmosphere, and this initiates the free radical polymerization at the reactive sites. With this plasma-initiated graft polymerization technique, a desired polymer can be covalently bound to the surface of our material, resulting in a nanocoating or a grafted biomaterial. These nanocoatings can be synthesized with controllable polymer density and controllable polymer brush lengths. The grafting density for these surfaces was calculated to be 48 micrograms per centimeter squared, and this was done using TBO staining, which is toluidine blue O. The polymer brush lengths were determined to be 55 nanometers according to AFM imaging, which I will show you in a bit. In the final step of the fabrication process, silver nanoparticles are immobilized onto the surfaces, creating anti-infection biopolymers. And this is done by immersing our samples into 2 mLs of 5 millimolar stock solutions of our silver nanoparticles for 30 minutes. This results in a immobilization concentration of 9.75 micrograms per centimeter squared. So this tells us the concentration of silver nanoparticles on our surfaces. It should be stated that this concentration is significantly lower than any other anti-infectious surfaces in literature. The construction of anti-infection biomaterials essentially involved 3 steps. The first involves the treatment of either PET or silica wafers to 80 watts of argon plasma for 3 minutes. The second is the plasma-initiated surface polymerization reaction. And the third involves the immobilization of silver nanoparticles. The immobilization of silver nanoparticles occurred naturally and through electrostatic interactions. This is because of the negative charge on the grafted surfaces from the deprotonation of carboxylic acid groups in solutions with pHs of higher than 4.5, and the positive charge on silver nanoparticles. Each of these different modification steps were visualized using SEM. Image 1 is our plasma exposure. Image 2 represents our polymer grafted surface. And SEM image 3 represents our immobilized surface. Here is the chemical structure of acrylic acid. This is the monomer used for the construction of our grafted surfaces. Figure 1 shows ATR, FTIR spectrum of our plasma-treated biomaterials, also known as our control, and the acrylic acid grafted surface. Figure 2 shows our control group, our acrylic acid surface, our plasma-treated surface, and our anti-infectious surfaces, also known as the acrylic acid grafted surface with silver nanoparticles. But back to figure 1. As we can see, there is clear evidence of acrylic acid bound to our surfaces. How do we know that? Well, by the presence of the carboxylic peak around 3400 wave number. And this is accompanied by another peak, a carbon hydrogen peak around 2850 wave number. And let's not forget about our carbonyl group at 1780 wave number. When we combine all of these these peaks, we get the chemical structure of acrylic acid. However, when we go to image 2 or figure 2, we see that there's a difference between figure 1 and figure 2, the red line and the blue line. The reason is figure 2 has our silver nanoparticles bound to the surface. And because they're bound to the surface, they decrease the intensity of our carboxylic acid group. And interestingly enough, they increase the intensity of our carbon-hydrogen peak. Now, if we just focus on these two peaks, we see that there's other differences. And it's not just based off of the intensity. For example, our carboxylic acid group, it shifted. It shifted from 3,400 in our figure 1 to 3,300 in figure 2. Also, the carbon-hydrogen peaks shifted. It shifted from 1850 to 1750. And the reason becomes why? Well, the reason is because the silver nanoparticles are immobilized onto the surface, and this interferes with the chemical composition. It's more of a tweak of our acrylic acid surfaces. Another interesting factor is that we see carbonyl peaks that are less intense, and they shifted from 1780 from figure 1. It shifted down to 1650 in figure 2. This also tells us something very interesting. It states, or it allows us to think that the silver nanoparticles are bound to the acrylic acid or the carbonyl and the carboxylic acid group of our acrylic acid polymers. As expected, each modification step was accompanied by substantial surface topography changes, and this was visualized using AFM and corresponding SEM imaging. After each modification step, the physical properties of the surface changed. When analyzing plasma-treated PET films, these films had no polymer brush thickness or polymer brush density. And this is demonstrated in both AFM and SEM imaging. We use these plasma-treated films as our baseline. These samples were then compared to the acrylic acid PET film. And we see that the surface changed from 0 nanometers to 55 nanometers. And we saw that our surfaces became a little bit more rough in AFM imaging. On our immobilized surfaces, not only did the thickness of the polymer brushes increase, we see that our surfaces became even more rough. And the reason is simply because of the immobilization of our silver nanoparticles. Immobilized materials, in comparison to other materials, caused bacterial cell death in MRSA, S. epidermidis, and E. coli biofilm forming bacteria. The anti-infection activity of the immobilized surfaces was evaluated and tested by loading 3 different strains of bacteria: S. aureus, S. epidermidis, and E. coli. Our contaminated surfaces were incubated under stationary conditions at 37 degrees Celsius in TSBG media. The y-axis represents the absorbance from live bacteria, while the x-axis represents different types of biomaterials used in this study. At the end of the overnight culture, live bacteria in biofilms grew on different biomaterials, and this was quantified using MTT assay. The MTT assay is a viability assay, and it quantitatively measures the metabolic activity of cells. Viable bacterial numbers on surfaces were quantified using optical density at 570 nanometers. These changes allowed us to compare live bacteria versus dead bacteria. The data represents the mean and standard deviation of at least 20 samples. The data obtained in this study suggests that our control surfaces, and what I mean by control surfaces are our plasma treated surfaces and our grafted surfaces, showed negligible amounts of antibacterial activity. On the other hand, our immobilized surfaces show or demonstrate significant antibacterial activity as stated or as shown by the very low absorbance readings. SEM images, along with corresponding confocal microscopy images of S. aureus, S. epidermidis, and E. coli bacteria cultured on grafted surfaces and immobilized surfaces for 18 hours were taken and were useful in confirming the results in the MTT staining assay. The scale bar for SEM images is 1 micron, and the scale bar for confocal microscopy images is 20 microns. SEM images on grafted surfaces contaminated with bacteria overnight show healthy, round, and full-shaped bacteria, demonstrating the intermediate stages of biofilm formation. On the other hand, bacteria that were cultured or were attached to immobilized surfaces show dead bacteria. And the way how we can determine it is through the crevices and how it looks like a raisin on each different strain of bacteria. They're deformed and they're unsymmetrical in shape. Confocal microscopy images and the live dead staining kit was used to further evaluate the antibacterial activity of our surfaces. The Cyto 9 dye of the live dead staining kit is a nucleic probe with green fluorescent color and is permeable to healthy cell membranes. Thus, it only stains live and healthy cells. Propidium iodide of the live dead kit is a nucleic probe with red fluorescent color. And this is only permeable to dead cells. So it only stains dead cells red. In other words, the green color that we're seeing on our grafted surfaces indicate live, healthy bacteria. On the other hand, on immobilized surfaces, the red color that we all see demonstrates dead bacteria. Due to the excellent anti-infection activity of our immobilized surfaces, it was imperative for us to test the biocompatibility of these same surfaces and evaluate the results. The conditions of human fetal osteoblasts were monitored using, once again, confocal microscopy and the live dead staining kit. Remember, green indicates living cells, red indicates dead cells. So we grew human fetal osteoblasts on plasma treated films, which was our control, acrylic acid grafted films, and immobilized surfaces. Once again, the cells grown on our plasma treated PET films was taken as 100% and everything was compared to that. The scale bar is 50 microns. Results from this study demonstrate the biocompatibility of all 3 biomaterials. And once again, this is indicated by the green color that we see. The human fetal osteoblasts cultured on our grafted surfaces grew healthy and rapidly. And unlike bacteria, human fetal osteoblasts cultured on immobilized surfaces were healthy and grew steady throughout the time span of the 7 days. Confocal microscopy also revealed that our immobilized surfaces allowed for proliferation and continuous cell growth. The results obtained from these studies suggest that immobilized surfaces possess a balance between killing and biocompatibility of tissue cells. Specificity studies involving human fetal osteoblasts and S. aureus bacteria grown for 5 days on acrylic acid grafted surfaces and immobilized surfaces were monitored using the live dead staining kit. Once again, the scale bar is 50 microns. In order to examine the specificity of immobilized surfaces in vitro, co-cultures involving S. aureus and human fetal cells were performed on grafted surfaces without nanoparticles and on immobilized surfaces. First, both the grafted surface and the immobilized surface were exposed to bacteria for 10 hours. Next, the contaminated biomaterials were washed with PBS and tissue cells were seeded on both materials for an additional 10 hours. Then the biomaterials were washed and cultured for 5 days. This experimental procedure was used to model the contamination of biomaterials and medical device post-implantation. When grafted surfaces containing live bacteria were exposed to tissue cells, the live adherent bacteria grew continuously and drastically modified their environment. As the bacteria flourished, they lowered the pH of the culture media, creating a toxic environment for osteoblasts. In other words, we see live bacteria and dead tissue cells on grafted surfaces. Unlike the grafted surfaces, immobilized surfaces killed the adherent bacteria and allowed for tissue cells to thrive. Tissue cells on immobilized surfaces were healthy and viable, allowing for proliferation. In order to study the mechanism of action of our anti-infection surfaces, the release profile of silver nanoparticles from immobilized surfaces was monitored using inductively coupled plasma optical emission spectroscopy. The y-axis on both graphs represent the concentration of silver nanoparticles released from surfaces. Graph A represents exposure of our immobilized surfaces to either 2% fetal bovine serum or simulated body fluid. And this was all done for 72 hours. Graph B represents exposure of immobilized surfaces to solutions of PBS with different pHs ranging from 7.4 to 4.5. This data represent the mean and standard deviation of at least 20 samples. When testing the stability of our immobilized samples, it appears that salts from simulated body fluid and proteins, sugars, and biomacromolecules from the fetal bovine serum did not interfere with the interaction of the nanocoating and the silver nanoparticles. In other words, there was no trigger of release of silver nanoparticles from the grafted surface. In fact, the surfaces were extremely stable, allowing for less than 10% of silver nanoparticle release after the 72 hours of exposure. As demonstrated through the work of others, adherent bacteria drop the local pH of solutions and of their microenvironment when colonizing a surface. In order to better understand the chemical effects of pH on our surfaces, we monitored the release of silver nanoparticles from immobilized surfaces in PBS with various pHs. Contrary to the work of others, our immobilized surfaces were stable when placed in these wide range of pH solutions for 72 hours. We can confidently say that less than 10% of silver nanoparticles were released from our surfaces into the supernatant. Therefore, we can say that there is a strong affinity between our surfaces and the silver nanoparticles. Scanning electron microscopy was used to further visualize our surfaces, but at the nano level. The scale bar for these images 1 and 2, is 400 nanometers. In these images, grafted surfaces, image 1, possessed densities of 48 micrograms per centimeter squared, and these coatings demonstrated uniform polymer brush coverage. Immobilized surfaces, or image 2, demonstrated evenly dispersed silver nanoparticles throughout the surface of the grafted material. In order to even get a better understanding of the physical properties of our surfaces, even highly or more highly magnified SEM images of the nanocoatings were taken and were studied. Image A is a magnified image of image 1. Image B is a magnified image of image 2, and the scale bar for image A and B is 100 nanometers. Image C is an even more magnified image of image B, and the scale bar is 50 nanometers. So let's go back to image 1. Once again, image 1 demonstrates uniform polymer brush coverage with no cross-linking. However, as silver nanoparticles were immobilized onto the grafted surfaces, and as seen in image 2, multiple attachment sites between the polymer brushes and the silver nanoparticles are apparent and were confirmed in ATR, FTIR peak shifts. In addition, the high stability of the anti-infection surfaces from our previous slide can therefore be explained by the multiple attachment sites between the polymer brushes and the silver nanoparticles, resulting in these cross-linked nanocoatings. When immobilized surfaces were further examined, multi-layer cross-linked silver nanoparticles surfaces were constructed under the above experimental conditions. Silver nanoparticles uniformly distributed throughout the surface and were bound in between the polymer brushes in a multi-layer fashion, and there was minimal silver nanoparticle aggregation. This form of immobilization, rather than the deposition of silver nanoparticles specifically located at the surface of the modified material, allows for an even distribution and an appropriate concentration for antibacterial activity. When compiling all of our data, and more specifically, the data from stability assays, the anti-infection biomaterials constructed in this work did not have a gradual or a triggered silver nanoparticle release mechanism. Therefore, a different mechanism of action should be proposed. It is postulated that silver nanoparticles immobilized onto our surfaces bind to the cell walls of adherent bacteria. This eventually results in the penetration of the silver nanoparticles into the cell membrane, causing structural damage and eventual cell death. Acrylic acid grafted surfaces were constructed as a platform for the mobilization of silver nanoparticles in our study. When generated under appropriate experimental conditions, silver nanoparticles were cross-linked and formed cross-linked nanocoatings and could kill bacteria indiscriminately at the site of adherence without releasing silver nanoparticles. Under our optimal grafting conditions, the surfaces constructed in this study were capable of effectively inhibiting both gram-positive and gram-negative biofilm forming bacteria. The surfaces also demonstrated excellent specificity, targeting infectious organisms efficiently and effectively. Several attractive advantages of these surfaces include their broad spectrum of activity against antibiotic-resistant bacteria. 2, the unlikelihood for bacterial resistance due to their proposed mechanism. And 3, specificity. Our surfaces could distinguish between tissue cells and microorganisms. And they were biocompatible and they were stable. Some of our future direction involves understanding the reasons why our surfaces are in fact selective. We want to improve our biocompatibility, and we want to study the longevity of our surfaces. These are a couple of publications from this particular work, and these were some of the students that actually worked on these 2 projects. Thank you so much, and I'll take questions.